Cannibals like to say, when the food runs out, at least we have each other. It's the same with esters. When esters form enolates, if there's no better electrophile around, they will find another ester molecule that hasn't yet enolized. That is the focus of this chapter, reactions of ester enolates with esters. It's important to be able to look at an ester and identify the electrophilic center or centers if it's conjugated, and similarly identify the nucleophilic centers in an enolate or a conjugated enolate. Taking the simple case of ethyl acetate reacting with itself in the presence of ethoxide, formation of an enolate generates a nucleophile that adds to a second ester molecule. This gives a tetrahedral intermediate, leading to expulsion of ethoxide and formation of a ketoester. The ketoester formed in this process is more acidic than ethyl acetate, so it will be deprotonated preferentially. The reaction will rest as that stable anion, even though each step is reversible, and more than one equivalent of base will be required. These intermolecular reactions work extremely well if the two esters involved are the same. So it's important to be able to look at ketoesters and recognize how they might be made by this type of condensation. To do this, check to see if there's a bond that may be formed by reacting an ester enolate with the same ester molecule. Using the same alkoxide base that comprises the ester is good practice in these reactions to avoid transesterification. Heterocoupling between two different esters is useful for some combinations, but only some. If both esters are enolizable, then 4 different products may form, and the reaction is so unselective that it's virtually useless. Heterocouplings are useful, however, if one of the esters is non-enolizable. In this first example, the non-enolizable compound, that's diethyl carbonate, is actually not an ester, but it's still a good electrophile in these condensation reactions. Similarly, methyl benzoate can be used to condense with enolizable esters. Intramolecular condensations of enolates with esters is an important basis for cyclization reactions. It helps to number the carbons between the two carbonyl groups. So in this case, the enolate formed at C5 will react with the carbonyl C1 to form a 5-membered ring. In the second case here, the enolate is not formed from an ester, it's formed from a ketone, but the same type of reaction occurs. That ketone enolate attacks the carbonyl of the ester to expel methoxide and form a 6-membered ring. The only alternative would be to form a 4-membered ring fused to the benzene, and that's clearly not desirable. In this third example, again, a 5-membered ring is formed, and in the fourth one, a spirocycle. Those examples selected above are simple because the two ester ends of the molecule are identical. They're related by symmetry. If the diester starting material is not symmetrical, then 2 different rings may form, and the reaction will only be selective if one ring size is more favorable than the other. Let's suppose that we take products of these condensation reactions and hydrolyze the ester under basic conditions. That gives carboxylates, which have a very nice electron flow pathway to decompose to enolates with the liberation of carbon dioxide. Similar pathways are available even if the other group is not a ketone. For instance, it could be a nitrile or a nitro compound. If instead ketoesters are hydrolyzed using acid, then this gives carboxylic acids. And they have a different pathway to push electron flow to enols and CO2. Bye-bye.